we are going to study about glycolysis, gluconeogenesis, and glycogenolysis. Okay, so first, what is glycolysis? It's the breakdown of glucose to become 2-pyruvate. Okay, in simple word, we can say like that, and that doing fed state. That means after eating food, fed state glycolysis happen. So, how does this happen? So after taking food, the glucose from circulation it will enter liver. Okay. So there is a GLUT2 transporter, which is two-way transporter, which captures this glucose with 6 carbon and put it inside the liver. Okay. So this GLUT2 transporter can even take it back and put to the circulation. So in that case, there is an enzyme called glucokinase. Kinase means enzyme. Okay. The purpose of this glucokinase enzyme is to add a phosphate in the 6 carbon. Okay. So it will become the name change. So this GLUT2 transporter will not identify it as just glucose. Okay. So it will not take it back. But there is another enzyme which removes this phosphate, that is glucose 6 phosphatase. Okay, so here already this phosphate is added. In some cases this glucokinase enzyme is not present when we are young, like that, newborn, like that time. So that disease or that problem is known as MODY, maturity onset diabetes of young people. Okay, so now this is converted to glucose 6 phosphate, means one phosphate is added to the 6th carbon. From there it is again converted to fructose 6 phosphate, then fructose 1 6 bisphosphate. Y means 2 and S is separate. So here 1 and 6 is written. So the first and 6th carbon has phosphate. Then it is again converted to glycerol aldehyde 3 phosphate. Third carbon will have the phosphate, and the other side is dihydroxyacetone phosphate. Everything is same, just the name is different. Okay, so here also in the third position phosphate came, and again it is converted to 1,3 bis phosphoglycerate. Both sides same, and then comes 3 phosphoglycerate, then 2 phosphoglycerate, and PEP, phosphoenol pyruvate, and finally pyruvate, 2 pyruvate. So this is the total process from of glycolysis, glucose to pyruvate, and later it is converted to acetyl CoA, and it will go to Krebs cycle to make ATP for quick energy. And the next one is gluconeogenesis. It is written breakdown of non-carbon structure in fasting more than 6 hours. So if you want to do the fat loss, we have to do this. Okay, so we fast for 6 hours and do the workout. Let's see what happens. So in the gluconeogenesis, when we eat, actually we eat food which is extra, means we eat food for our energy, and more glucose what we are eating is stored in our liver and muscle. Okay, in the liver it is stored in hepatic cell. Okay, and 80 to 100 gram of glucose is stored in liver, it is glycogen, and also in muscle 250 to 400 gram is stored as glycogen. So whenever our means 6 hours fasting, our ATP is depleting. That time, first the body will take this stored glycogen from the liver for energy. Okay, so this process is known as glycogenolysis. Okay, so after what happens, our 80 gram, 100 gram, this glycogen is also finished. Then the body starts. The body will start to take glycogen from the muscle. But here the problem is it doesn't have this glucose 6 phosphatase. Means here we have that to remove the phosphate. So it has to break down this glycogen into pyruvate, and this pyruvate along with there is a subproduct in this process, that is lactate and alanine. So this pyruvate along with lactate and alanine will go to the liver. Means it's the decision of liver to whether it take it to ATP, means acetyl CoA and go through Krebs cycle, or take it back the reverse process to form glucose. Okay. And if you are working out and you want to burn the fat, first 27 minutes you do high intensity workout only, your first glucose and stored glycogen will be finished. Only then here this happens. Means the fat is stored as triglycerol in our body, and this triglycerol has fatty acids and glycerol bond, and it gets separated, and this glycerol bond will go to this area, this glycerol aldehyde. It will become glycerol aldehyde by the process of this glycerol kinase. That enzyme will make it this glycerol bond into glycerol aldehyde, and it will go all the way back and brings glucose.